Computation and System Biology. Assignment help. For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingdassignmenthelp.com, info@nursingdassignmenthelp.com, or reach us at https://www.nursingassignmenthelp.com. Protein-protein interactions are often stored in databases that cover thousands of proteins and interactions between them. However, there are biases present in these data. Poorly studied proteins may be underrepresented in these databases because few people have taken the time to identify interacting partners. Thus, the databases are overrepresented among highly studied proteins. Evidence of protein-protein interaction is highly variable, as there exist diverse biochemical assays to identify protein-protein interaction, and these assays in themselves are biased towards specific types of proteins. In this problem, we will study these biases and the relationship between them. You will be completing the script citationNetwork.py using the NetworkX module, which allows us to manipulate and study networks in Python. NetworkX has been installed on Athena, so we will follow a similar procedure as other problems. Log on to Athena's dial-up service. SSH your Kerberos username at athena.dialup.mit.edu. Before running any Python scripts, use the following command to add the NetworkX module we installed to your Python path. Export Python path equals /afs/athena/course/20/20.320/pythonlib/python 2.7/site packages/. Assessing bias in high-throughput protein-protein interaction networks. Nursingassignmenthelp.com. Otherwise, you will get an importer. You will also need to get the .zip containing the files for this problem in the course folder. cp/afs/athena/course/7/7.91/sp_2014/citation network.zip cd unzip citation network.zip cd citation network. Please submit the citationNetwork.py script online. It should take a uniprot file and a network and print some scaffold text. Of course, feel free to modify the script in any way that is helpful to you to answer the questions, but have it conform to the above standard when you submit it. Bias in protein studies. In the zip file, we have provided protein citation data from uniprot. This file was generated using the following procedure. You do not have to generate the file. Search to select only those proteins that are human and their status is reviewed. Click on Customize to make sure the table has only the mapped PubMed ID field, and then download the tab delimited file. In citationNetwork.py, for each unique entry name in this file, collect the unique number of mapped PubMed ID, correlating to the number of times the protein has been cited. Nursingassignmenthelp.com. Evidence description neighborhood score computed from the intergene nucleotide count fusion score derived from fused proteins in other species co-occurrence score of the phyletic profile, derived from similar absence/presence of genes, co-expression score derived from similar pattern of mRNA expression measured by DNA arrays, and similar technologies experimental score derived from experimental data, such as affinity chromatography database score derived from curated data of various databases text mining score derived from the co-occurrence of gene/protein names in abstracts. Plot a histogram of the number of citations per protein. Attach the PDF to this write-up. B. What are the median citation rate and maximum citation rate? Median, 3, maximum, 52-56. C. Which protein has been cited the most? B. Bias in source of interaction evidence. String is a database of protein-protein interactions with confidence scores ascribed to each interaction based on distinct sources of scoring contribution. For each source of evidence, we've provided you with a .pkl file containing a distinct NetworkX graph with the proteins, nodes, and interactions, edges, between them. The weight of the edge is the normalized score for that particular source of evidence, if it was greater than 0.25. You will find a function to load these, and directions for interacting with them in the script. Nursingassignmenthelp.com. How many edges, interactions, and nodes, proteins, are in each protein interaction network? Network edges nodes neighborhood score 19976, 1207 fusion score 296, 240 co-occurrence score 3944 1148 co-expression score 149333, 3967. Cutoff equals 0.4. Network edges nodes neighborhood score 8354 866 fusion score 144 118 co-occurrence score 1110 550 co-expression score 49874 2390 experimental score 95245 9772 database score 233910 6422 text mining score 620,858 14481. Cutoff equals 0.8. Network edges nodes neighborhood score 412 154 fusion score 1816 co-occurrence score 00 co-expression score 5172 642 experimental score 16523 4035 database score 233910 6422 text mining score 89560 9416. NursingAssignmentHelp.com. C. Comment on what these results say about the different types of evidence. Many of the data types have a low proportion of high confidence interactions, indicating that much of the data may be of low quality. As long as a comment on data quality or reliability was made, points were awarded. In many cases, students commented on specific types of evidence, which was also accepted. C. Relationship between number of citations and node degree. The size and distribution of an interaction network measured by distinct types of evidence varies greatly. For each interaction network, collect the node degree of each protein. Then, after removing proteins without interactions and interacting nodes without data in UniProt, calculate the Spearman rank correlation to determine if the node degree is correlated with the number of citations of that protein collected in the first section. A. What is the node citation/interaction correlation for each of the sources of evidence? B. What are the correlation values when you restrict the interactions to those with at least a score of 0.4? 0.8? Network no cutoff 0.4 0.8 neighborhood score 0.058 0.075 fusion score 0.104 0.182 0.370 co-occurrence score 0.017 minus 0.002 co-expression score 0.008 0.012 0.051 experimental score 0.416 0.429 0.247 database score 0.104 0.104 0.034 text mining score 0.706 0.685 0.564. NursingAssignmentHelp.com. C. Is there a relationship between the number of citations and degree? If so, what is the relationship and why do you think this is? For some forms of evidence, there is a correlation between number of citations and edge degree. This may be due to bias in how much certain proteins have been studied. The more a protein has been experimented on, the more true interactions and false positive are likely to be discovered. Also, if a protein has lots of connections, it is likely to be mentioned in the context of its partners. Many people said there was a positive correlation, despite the fact that in many cases it was very low. It was quite open to interpretation, depending on what people considered a strong correlation. D. Does this relationship vary between sources of evidence? If so, why? Yes, fusion, experimental, and text mining score have higher correlations than the other data types. This is likely because the data for these experiments comes from a large pool of smaller studies, which have bias in choice of protein to study. Genome-wide assays that look at all proteins at once do not have this problem. Again, this was open to interpretation. To copy your script and histogram from Athena onto your own computer, use SCP. scpr@athena.dialup.mit.edu/citation network/citation network.py scpr@athena.dialup.mit.edu/citation network/histogram.pdf NursingAssignmentHelp.com. B. The C, M, T, and J variables in the segue model implement a countdown function, one of the core features of segue. How might these countdown variables improve on a simple HMM model in modeling the underlying genomic states? A traditional HMM doesn't allow tuning for state duration. In the case where we train with a large number of labels, we might expect results to incomprehensible with frequent switching between